Hey guys, so up to this point, you've gone through DNA, DNA replication, RNA, and protein synthesis. So that's transcription, translation. You've already done work with transcription, which is moving from DNA to an mRNA, and you've already worked with translation, which is moving from that mRNA and coding it into a protein. Now, hopefully you remember all of those where they take place. And next, what we're going to do is we're going to apply all of protein synthesis together. We've done it individually, but now we're going to put it all together. With a project, we're going to identify a fictional organism. We're going to look at the snork. These snorks have one chromosome that have 10 genes located on said chromosome. Now, what you're going to do is you're going to look through 4 different snorks' DNA, and then you're going to make the mRNA as well as the protein that goes along with that gene. And from that, you're actually going to give an entire phenotype, and you're going to be able to draw a snork. Now, what I've given you is you have an entire chart that composes of all the different genes that are located on these chromosomes. So each of them code for something else. They code for something in particular. It tells you what the proteins, the amino acid sequence should be, and then it gives you what the phenotype is. Each gene has 2 different phenotypes possible. As you know, humans, we've got lots of different phenotypes, so this is very, very scaled down to help you out there. So you have 4 different snorks, okay? So what you're going to do is you're going to take their DNA and you're going to turn it into mRNA and then to a protein. Now, the guy that we're going to work with is the Snicker snork, okay? Now, I've already made it easier to where it's broken into groups of 3. Remember, mRNA needs to be broken into groups of 3. Those are called codons. Codons give rise to one amino acid. The DNA is already broken up like that for you. Now, what you need to pay attention to is, remember, there are 3 stop codons and there is 1 start codon. Each gene will start with a start, as you would assume, and will end with a stop. Now, the only time that we will continue to transcribe or make mRNA is when we have a new start codon. So together, I'm going to go over one of these rows for us. Now, do not think that this row is the end of the chromosome. This is the entire DNA sequence. So all of this is one sequence. It doesn't break up. They just continue to the next row. Okay? So what we're going to do is let's use red for our mRNA. So you need to go ahead and code for this DNA strand. I'm going to make sure you use your base pairing rules for mRNA, well, RNA in general. So T goes with A goes with U, C goes with G, leave a space. C goes with G, A goes with U, G goes with C. Okay, and then we just continue doing that until we finish the entire row. Okay, G, C, G, A, U, G, UAC, CCC, CAA, CAG, AAA, UAG, and then UA. Okay, so that is that first row. Now, we would continue to keep going through. Now, I'm not going to do that on this video. I'm only going to give you, the beginning here to help you out. Okay, so there's our mRNA right there. Now, you need to remember that what your start codon is. Remember, if you don't remember, I'm going to write these in blue. Your start codon is always going to be AUG, and it's going to be MET, okay, which is methionine. Now, you will need your lovely codon charts with you to complete this. I'm using the box one. I prefer the circle one, but the box one is a lot easier to go through. So the first one, AUG, is going to be met, MET. So AUG, met. Okay. Now, the second sequence is GUC. So GUC, which is Val. Yay. Woo! VA. Oh, no. VAL. Okay. Next is AGC. So let's do AGC. ARG. ARG. AAA. A. Lys. LYS. And then UAA. UAA is a stop codon. So stop. Now, that would be the first gene. Okay. So this right here, let me go ahead and we can highlight in yellow. This is gene number 1. Okay. Gene number 1. Now, remember, we do not transcribe if there is not a start codon. So is GCG, is that a start codon? No. The answer is no. So we do not have to do that. So what I'm going to do is I'm going to put 3 X's so I know that there's no amino acid there. Now, AUG, that is our start codon, so we put met. Okay. Next is UAC. UAC, tyrosine. CCC, CC and C is proline. CAA, CAA. GLN, glycine. CAG, CAG, glycine again. AAA, AAA, glycine. UAG, UAG. Stop. Okay. Now, next is, that's going to be gene number 2 right here. So this is gene number 2. Let's tab over. A little space, number 2. There's gene 2. Okay. Now remember, we don't start transcribing until we get another start. Okay. So again, UUA, that's not a start codon, so we would not make that into an amino acid. Again, you're going to continue to go through the entire thing. Picture this as one strand. Do not break it up like I'm doing right now. Okay, so you would continue to go all the way through. Now, that's going to be what we have so far. So now we need to go and figure out what that gene means. So our gene is Val, Arg, Lys. Okay, so we come up here and we look at number 1, and we don't have the right thing, right? So we need to look back and double check and make sure we did this correctly. So TCG is AGC, AGC, which was Sayre. There we go. Messed that up. Okay. So Val, Sayre, Lys. Val, Sayre, Lys. So this guy is going to be Harry. Poor guy. All right. Let's look at number 2. Let's look at our next traits. Tyrosine, pro, GG, Lys. Go to number 2. It starts with tyrosine, pro, and he's going to be plump. Okay. Now, once you do the entire strand, you'll come down here. You'll see that there's already a spot asking for his phenotype. So we have, so far, he is going to be hairy, plump. Now, you'll need to give me all 10 as you go, okay? Now, if that doesn't help clear it up, you can email me. We can do a Google Meet. We can talk this through. Just let me know what else I can do to help you. But good luck, and I'm looking forward to seeing the pictures and seeing what they look like. Usually these have been really good in the past. I'd like to keep that up. You will have to draw your snorkel on paper, take a picture, and upload it like you have been. Just as you go, any questions, just let me know.